We'll dive deeper into the role of microbial volatiles to volatiles, particularly in how plants respond to them through specific proteins context.
Okay, thank you for the introduction. It's a pleasure to be here presenting a part of result of my thesis. Today, I'm going to talk about CLPC2. Microvolatile responsive chaperone is an important determinant of plant growth, development, and photosynthesis. And Rafa explained before, beneficial pathogen microorganisms promote the growth, the photosynthesis, and produce changes in the photosynthesis machinery, producing the plant more sugar and isoprenoid like carotenoid, hormone, and photosynthesis pigment. And this compound act like a signal, producing changes in the expression of protein involved in chloroplast proteostasis, protein like a chaperone, like CLP complex. The CLP complex has a client or target that are located in the different steps of the photosynthesis. Okay, this is a proteomic analysis of plant exposed to microbial volatile, and we can see here a lot of protein that are client or target from the CLP complex, including an overaccumulation of the CLPC2, part of the CLP complex. The CLP complex is formed part of the chloroplastic proteostasis machinery, formed by adaptation unity, CLPS1 and CLPF, that mark the misfold protein. This marked protein is recognized by the CLP complex formed by CLPC1, CLPC2, and CLPD, and processed by the protease core. Okay, we know that CLPC1 regulate different protein involved in isoprenoid biosynthesis enzyme, the same enzyme that appear in the proteomic analysis of plant exposed to microbial volatile. But does CLPC2 play an important role in plant growth, development, and response to volatile compound? Okay, there are two different opinions. Previous paper described that CLPC1 and CLPC2 have redundant function, and recently appeared a publication published that CLPC1 and CLPC2 have functional specificity. First, we compare proteomic analysis that we did in our lab of CLPC2 versus wild type with with a proteomic analysis of CLPC1. We can see here that the two proteomic profiles are totally different, and only two proteins appear in the both proteomic profile. In addition, we compare the phenotype of plant CLPC2 mutant and CLPC1 mutant, and here we can see that CLPC2 and CLPC1 has a smaller respect to wild type, and CLPC2 mutant is darker respect to wild type, and CLPC1 has a yellowish phenotype. When analyze the photosynthesis, we discover that CLPC2 has a lower photosynthesis, stomatal aperture, and transpiration. When we analyze the seed development, we saw that CLPC2 seeds have a wrinkle phenotype and lower specific per weight, a lower total fame. And the yield, we analyze the yield and compare with the wild type, We discovered that has a lower number of elongation inflorescences per plant, the lower number of silk per plant, less seed yield. We also analyze the hormone homeostasis and detect or discover that CLPC2 mutant overaccumulate ABA, salicylic acid, hormonic acid, and produce less indolacetic acid and cytokinins. It explains the reduced stomatal aperture, the low photosynthesis, and the low number of inflorescences. The big question was CLPC2 respond to volatile compound. Okay, we analyzed the growth promotion of plant of CLPC2 exposed to microbial volatile, and we discovered that CLPC2 doesn't respond to volatile compound if we compare with the wild type and CLPC2 with complemented. What is the action mechanism of CLPC2? You probably remember this proteomic analysis of plant exposed to microbial volatile, and the red mark protein are proteins that appear or are client of CLPC1, but in this proteomic analysis didn't appear CLPC1, appear CLPC2. So we we made plant with wild type with an overexpression of CLPC2 and discovered that act like a CLPC1 in phenotype with a yellowish phenotype. When we analyze the concentration of protein that are target from CLPC1, we discovered that have a similar profile in CLPC1 wild type with overexpression on CLPC2 and totally different respect to CLPC2 mutant. With overall data, we suggested a model action of CLPC2 in wild type plant in normal condition. CLPC1 recognize the client of CLPC1 and are processed by the protease core. In CLPC1 mutant, in this condition don't exist this CLPC1 ring, so the protein that are client of CLPC1 are overaccumulating in the chloroplast, and in condition like CLPC1 or high expression of CLPC2 or volatile compound exposure, CLPC2 is in the same site like CLPC1. CLPC2 ring doesn't recognize the mis4 protein that are client of CLPC1 and overaccumulate the same protein like in the CLPC1 mutant. But in this model, CLPC2 act like CLPC2 and recognize the own protein of CLPC2 and processes. Other suggested model that it could be a good option is that CLPC2 act like a cap blocking CLPC1 ring and produce an accumulation of mis4 protein that are client of CLPC1. So our conclusion is CLPC2 plays an important role in the response of plant to microbial volatile and in seed development. CLPC2 form part of the CLP complex and its interaction is different to that CLPC1. CLPC1 and CLPC2 are not redundant. They have different functional and molecular specificity. I have finished. So
No more. Sorry.
Okay, thank you. Any questions?
Thank you very much. And do you have some questions for the teacher?
CLPC1 mutant has a phenotype. We analyze other parameters that are not appearing here, but yes, this accumulation of misfold protein is a problem for the plant. Yeah.
And congratulations. I'm just very sorry. About the next step, because you have a lot of hypotheses in the end. Which is your next step in this project?
I would like to analyze the CLPC2 plants don't respond to microbial volatile. I would like to analyze if CLPC2 is involved in the immunity. So I would like to do any experiment, but I'm not sure if I have time to do it.
Thank you very much.